Okay, let's go and minimize that. Let's do that. Okay, and we're ready to go. So this particular one, this unit is called cellular biology, or I'm just going to put cell biology. All right, and that's what this particular first test is on. And again, I'm looking at that first note, and that first note was on something that we call cell theory. All right, and the first thing we talked about in terms of cell theory was what makes living things. So living things, and you'll notice when I'm creating my review notes, and this would have been the way I would have done it in university as well, I don't rewrite all the words, right? But what I write down are the keywords that I would think would show up in a multiple choice question that then I need to know about. So when it says they're living things, I'm talking about things that that living things all have. And so a lifespan. They have a lifespan. All right? What do I mean by a lifespan? What do I mean by a lifespan? Yep. That it lives and then ceases to live. Right? So how long it lives for, which means it's born and then it dies. Right? So living things all are born and then they die. They have growth, repair, and reproduce. Again, 3 terms, 3 words that you just need to remember. They grow, they're able to repair themselves, and they reproduce. They have senses. How do I spell senses? S? Sense. Is it an S or a C? Sense. Senses? Is that the way to do that? My spelling is terrible. I want to put a C in there. They can sense their environment, right? We can see, we can smell, all that type of stuff. They use energy, and they produce waste. All right? So all living things can undergo that type of stuff. And I'm just going to scroll down there a little bit. Okay. The next thing we did there, and I'll put here 2, was we talked about the microscope. Microscope. All right, we did a lot of work on the microscope, so being able to do some of those calculations are important, so we'll go over a few of those. Right now, I'm not going to go over it, but I'm going to tell you to see the handout for the parts. So, in my dream world, in the ideal world, when you go and sit down to, finish off the review, you're going to look at that picture and you're going to look at the parts. Because I might ask you a little bit about the parts and about those things. Okay. Next thing we talked about was magnification. And magnification is determined by those 2 lenses. What do I have to do with the values of the 2 lenses in order to get the magnification? Remember, there's the eyepiece or ocular lens, and then there's the lens at the bottom. How do I determine the magnification? I can see you checking your phone. Stop that. How do I determine the magnification when I have those 2 lenses? What did we do? No. How did we determine the magnification? We multiplied them. We multiplied those 2 things. So I take the ocular lens, or the eyepiece, it's called the ocular lens, ocular times the objective lens. All right? And an example there. The ocular lens tends to be times 10, right? That's what the ocular tends to be. The objective lens under low power. Does anybody remember what that objective lens was under low power? 4 times 4. Therefore, the magnification under low, what was that equal to? 40. So it would be 10 times 4 equals 40 times the magnification. Remember doing that type of stuff? What was the medium power? 10 and 10. So what was it? 100. And what was high power? 40. So 10 and 40. 400. Remember that? Okay. Actually, I'll finish those calculations here. I'll do magnification medium 10 times 10, 100, and magnification high 10 times 40, 400 times. Okay? And that's how we got that magnification. The other thing that we did was the diameter under high. All right? The other thing that we did was the diameter under high. So if I want to get the diameter, DH was equal to, anybody remember what the other things were in that equation? Had some magnifications, had some other diameters. If I had the H on the thing that I was looking for, what had to go on the top of the other one? The L's. So what were those other two? What were those other variables? Well, DH is what I'm looking for. So what's got to go on the top on the other side? ML, the magnification under low. What else goes with it? DL, the diameter under low. And what goes on the bottom? MH, the magnification under high. All right? Magnification under high. All right? Again, the D, I'm just going to put here, D stands for diameter. Right? That's how big that circle was. And the M stands for magnification. Okay? And we'll do a little example here just for a moment. Example. Let's say I have a DL of 4.5 millimeters. And let's say I have an MH, well, I might as well use my numbers, 400 times. And I have an ML of, well, what is it? 40 times. 40 times. Now, what did we first do with that DL? What did I want to do with that DL? I didn't like millimeters. So what did I turn that into? Anybody remember? Yep. Micrometers. And what did I have to do with that 45 to turn it into micrometers? Yep. Times by 1,000. 4,500 micrometers. Okay? Well, now we have everything that we need. DH is going to be ML 40 times DL 4,500 divided by MH 400, and you run that through a calculator and you get an answer. Okay? So we will need calculating devices for tomorrow. I'm not going to give you 5 or 10 of those questions, but I'll give you one. Right? There'll be one of those on that test. Okay? All right? I'll tell you right now, I will supply you with the equation. You just have to know how to get the information in there properly to get the answer. Okay? All right? And did anybody calculate that? Anybody got a calculator? Not a big deal. This is the same as actually dividing by 10 because the 40s kind of cancel away, so you're dividing it by 10, and the answer is 450 micrometers. That's how wide the view was. Okay? All right. One last thing I want to talk about before we move on from the microscope was, oops, that's a mistake. Can I erase? I determined I can erase this. Nice. So let's go back to this. Estimation. Let's say then that I'm looking at my microscope and I see that cell kind of looking like that. And I know that this width here, well, is 4,500 micrometers. What is the width of the object? Width of that object. What is the width of that? How did we do that? What did we have to do? Yep. Estimate how many fit across. So how many fit across, you think? 1, 2, 3, 4? 4? 4.5? Sit still. Try harder. Okay. 4.5. Let's go with 4. 4 fit across. So 450 micrometers, the width, divided by 4. If I go 4.5, it's nice and easy. We'll go 4.5. If I do 450 divided by 4.5, that is 100 micrometers. All right, so we have to estimate in order to determine that. Does that sound familiar to everybody? All right, excellent. Okay, so once we mastered the microscope, we again looked at cells and we looked at organelles. All right, we looked at the organelles within the cells, and organelles are essentially the cell parts with special functions. All right? Again, I need you to see your notes for the labels. I will be getting you to label cells. All right? You will be labeling the parts of the cells, and they're in that handout on organelles. So you'll need to go back and look at that entry on organelles. You would have wrote those organelles on that diagram, and that's where you're going to get it. If you didn't write them on the diagram, you could get it out of the textbook. The organelles would be in the textbook. You just got to find the page reference. I just want to talk a little bit about some of the organelles. Cytoplasm. What's the cytoplasm? What's the cytoplasm? Yep, Cade. The goo inside of the cell. It's the jelly-like substance that the other organelles are in. But you're right, it's the goo inside of the cell. I'm going to put jelly-like, and what do I have? Interior that holds other organelles. Interior that holds other organelles. So it's the stuff that's on the inside. Cell membrane. Where's the cell membrane? Where's the cell membrane? Yep. It's the outside. It's the structure that holds everything together. So it surrounds the cell and allows certain things in and out. It surrounds the cell and allows, 2 L's and allows substances in and out. What comes in to a cell? What's that? Nutrients. What else does a cell need? Oxygen. If I put O2, does that make sense? What leaves the cell? Waste. And what gases leave the cell? Yep. CO2. CO2. Excellent. The nucleus. What's found in the nucleus? The most important part of the nucleus. Yep. The DNA. The genetic information is found in the nucleus. It's the large sphere. All right. Genetic information found here. And we call that genetic information the DNA. All right. What does the nucleus do? It directs the cell what to do. All right? And we now know that when we say what to do, we mean, does it become a muscle cell? Is it behaving as a skin cell? Is it behaving as a bone cell? blood cell, etc., etc. Mitochondria. Mitochondria. What was the mitochondria known as? We had a little cell city analogy that I really didn't like it, but yep. Power plant. It is the power plant of the cell. It is the thing that produces the energy. Produces energy from food and O2. It uses the food and the O2 and it produces energy. It is the power plant of the cell. Vacuoles. Vacuoles are those big areas in the middle. They store food and water. They store food and water. All right, that's terrible water right there, water. And they can also store waste for removal. They can also store waste. Oops, I'm struggling with some spelling all of a sudden. They can also store waste. And we'll fix that water up since I'm here. Water. Okay? One special thing here, plants have a big vacuole. Plants have a big vacuole right in the center to store water. When it gets full, the plant stands up nice and tall. And when it uses all that water up and it hasn't had things watered, it then bends over because it loses that water inside that vacuole. All right? One last thing about these particular things, plant cells. So plants also have, so plants have some extras. They have a cell wall, all right? So they've got that extra layer. We can see that in our onion cells really nicely. When you zoomed right in, you could see there were two layers there. They have a cell wall. It supports the plant. It's what keeps it nice and tall. It supports the plant. And when we eat plants, that cell wall is known as the fiber. All right, you know how they say, get lots of fiber by eating your vegetables, right? That part of the plant that's chewy, you know, that is the cell wall. And they have something called chloroplasts. Chloroplasts. And the chloroplasts carry out photosynthesis. They're what's responsible for taking the sun's light and turning it into food. So chloroplasts carries out photosynthesis. All right? They make the food for the plants and ultimately for us. If we eat those plants, we're eating that food that they had made. Okay, we're moving on. So that's the end of our thing on organelles. All right? Oops, gone way too far away. So what number am I on? 4. Transit. Sorry, not transit. Transport. Transport in cells. We had a quick little note about how substances move in and out of cells. The first one was called diffusion. All right? And diffusion doesn't move water, but it moves stuff like salts and nutrients. So I'm going to just put moves, whoops, whoops, whoops. I'd like an undo. It'd be great if I had an undo button, but I don't. Moves, I'm just going to put here stuff because it's like nutrients and all that other thing. I know stuff is pretty vague, but it moves stuff in and out of cells. I don't mean water, and it always moves from high to low concentration. So it always moves from high to low concentration. We had a picture of it where I would have had a membrane here or something with lots of dots, just a few dots, and diffusion moves it in, right? High concentration slowly moves into where there's nothing. We saw that when we did our little lab with the little plastic bag. The iodine moved in. We saw that when we did our little experiment with those little cells made of agar. The sodium hydroxide moved in and turned it pink, right? We saw that movement in of a substance, and that's diffusion. The other one is osmosis, and osmosis is the movement of water, moves water in and out of cells, typically in, but it still moves water. Water goes in the opposite direction. The water will move to where there's a high concentration of salts. So it goes from the low concentration to the high concentration of salts. And it moves from a low to high concentration of salts. All right? You get lots of salt on your lips from eating sunflower seeds or eating popcorn, and the water comes out of those skin cells that are on your lips because it goes out to that high concentration salt, and your lips get dry. You put on lip stuff like that. Okay. All right. We're getting there. This is different than the optic stuff because there's not as much things that we're drawing. It's more information based. That's just the way biology is. 6, cells can't get too large because if they are too large and that nucleus is in the middle, the diffusion cannot get the food into the nucleus of the cell, so they would die. So cells have to divide when they get so big in order to prevent that from happening. And the next thing we talked about was cell division. Cell division. And we also had something called mitosis. That's the process that does that, okay? And at this point, again, I had you take some notes on a sheet. I'm going to just kind of draw it here. We have something called interphase, and then we had mitosis, and then we had cytokinesis, and the cycle just kept going round and round and round. All right? Interphase, that is normal cell life. Cell just doing its thing. Normal cell life. All right? Mitosis. Something gets doubled in mitosis. In the end, we get 2 of something. What gets doubled in mitosis? Yep. The nucleus and its genetic information gets doubled. Okay? So in mitosis, we are dividing, we are duplicating, duplicate the nucleus, I'm running into trouble in spelling, and the DNA. That's the most important part. It duplicates that DNA. If we started with 23 chromosomes in each of our cells, which we do as humans, when mitosis is done, there's 23 chromosomes, and everything's functioning, and everything is behaving properly. Cytokinesis. What happens during cytokinesis? What happens during cytokinesis? What is happening there? This cell actually splits, right? So once we've got the duplicate nucleus, we can split the cell. So this is the splitting of the cell, or splitting of the cytoplasm. Cytoplasm. Hence it's called cytokinesis. The cytoplasm is being cut. All right. The next thing we talked about in more detail was the actual mitosis. Let's talk about that. So under mitosis, There's some subdivisions we got to talk about. And the thing you're going to memorize is PMAT. All right? I don't know how that's a thing to remember, like a PMAT. I don't know what a PMAT would be. Like a mat that you... No, I don't know. That's weird, isn't it? Anyway, whatever helps you remember it, PMAT. And the P stood for the first phase. What was that first phase called? Yep. Prophase. Prophase. All right? And in prophase, we can see the chromosomes. So the DNA curls up and we can see the chromosomes. Can see the chromosomes. Can see chromosomes. And the nuclear membrane disappears. The nucleus starts to disintegrate. You can just see the chromosomes themselves, right? So nuclear membrane disappears. And if I'm drawing a picture of that, circle, I'm going to draw the nucleus as if it's kind of a dashed thing because the edge of it is starting to disappear. And I'm going to draw all these squiggly lines in here because we can start to see the chromosomes, right? Where before you couldn't tell it. It was just a ball, right? M. What's the M of PMAT? What's the M of PMAT? Logan? No idea. Exciting. It's going to be fun marking your test. Metaphase. 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 And what did the M of the metaphase help you remember? What happens in metaphase? Middle. What goes to the middle? Yep. The chromosomes. Chromosomes line up in the middle. Chromosomes line up in middle. Again, if I were to draw a picture of that, we would have our chromosomes, right? And they look kind of like X's that are kind of stuck together. The textbook had like 3 of them. And they drew these little lines kind of going to the end because there's these things that are starting to connect to them so that they can be pulled apart. But you can see there they're lined up in the middle. All right, that's how you would know. All right. A. What did the A stand for? What phase? Yep. Anaphase. Anaphase. All right. And I think of the A as being like anti. And if you're anti-something, you're opposite something. Your view is opposite to that. So the way I think of that is the chromosomes then are pulled to the opposite ends of the cell. Chromosomes. And they are pulled, but you can just say move to opposite ends. So again, if I'm drawing that one, now those little sister chromatids, right? We called them chromatids when they were separated out, are heading to the opposite side. But each chromatid is a duplicate pair of the genetic information. That's what happens kind of during interphase. Slowly it gets duplicated. Once it's duplicated, it says, okay, I'm ready to split now. And then the chromosomes present themselves and they pull apart. So we still have the same genetic information. And the T in PMAT, and I have really no way to memorize that one. Telophase or telophase, yep, telophase, telophase. And I don't know of a way to think of the beginning part, the telo, to memorize anything. It's just the end of it. A new nucleus is forming around the chromosomes and it's ready to break apart, right? So we have chromosomes uncurl and nuclear membrane forms. So the chromosomes, they disappear because they're uncurling, right? Kind of like a string uncurling. And we get a new nuclear membrane. In other words, the nucleus begins to form. So in this case, you've got kind of the dashed lines on either end representing that nucleus beginning to form, and those nuclei are starting to disappear, and we've got things doubled up. Now we just need cytokinesis to cut everything in half. And in the end, that's what I have here, cytokinesis. Cytokinesis. And the cytokinesis cuts in half. I'm just going to draw a picture for it. So in this case, I'm going to have like almost like a figure 8 type cell thing happening here, where now we've got the nucleus and the nucleus, and that thing is breaking apart, and we get our 2 cells. All right? Okay? And that is our cell division. Number 7. By the way, this is being recorded. I'm going to put that video on there. If you were so lazy that you couldn't possibly bother to read anything, you could re-listen to this. No? Make sense? You could increase the speed to like 2, make it go fast. You know what I'm saying? That could be your studying. Listening to me babble on. Yep.
Can you go to the YouTube? You have the videos?
They are linked. They're right on Google Classroom. All you got to do is click on Google Classroom. It'll move you to YouTube. It's this and my voice. It's the whole thing. It's the writing and my voice. All of those lessons are on there like that. Let's keep going. So next we talked about cancer. All right? And to be honest, we just made notes. Right? Remember, we made notes and there were definitions. I'm not going to write them all down right now. That was the other day. That wasn't too long ago. Go to your notes. All right? The title was Cell Cycle Gone Wrong. All right? And I'm just going to put here C notes. I want you to read through those notes. It had terms like tumor. What's a tumor? It had terms like benign versus malignant. All right? Remember, benign was the ones that were not cancerous, but malignant was the ones that were. It had terms like carcinogen, a substance that can cause cancer. All right? It had terms like mammogram, an X-ray that is used to test for breast cancer. So go through those notes, find that note, and just kind of read through those definitions. I don't want to go into great detail on that one. Number 8, this brings us to stem cells and specialized cells, which is what we did yesterday. Brings us right to yesterday. Okay, the cancer one was on Friday. So just a tiny little bit about stem cells and specialized cells. Stem cells. The term I used was undifferentiated. It's a crazy term, but it simply means cells that don't have a job yet. All right? So they're undifferentiated cells. Cells, no job yet. I don't know if I'd think of them as unemployed, because stem cells are, in terms of solving medical issues, they're golden. Like they're the best things ever, right? I wouldn't say that they have no job. They've got every possible job. Do you see the difference? They have the potential to do any job, right? It would be like the most employable person that could exist, right? So they don't have a job yet, but they can do every job, is the issue. That's what makes them so great. Types of stem cells: embryotic, EMB, I'm spelling trouble, embryotic. Embryotic, where do they come from? Well, they come from embryos. Embryos, those are that starting group of cells. As a human, very controversial, because in order to get the cells, you've prevented the development of a human, so that could be controversial. And then we also have tissue stem cells, and they can come from adults, which is good. That's a little less controversial, right? Take a few bone marrow cells from one human being. If you can find a match, put those bone marrow cells into another human being, and hopefully it treats their leukemia, and they can produce their own red blood cells, and things are good, right? That's the idea. And so they can come from places like bone marrow. Look at what does that say? Bone if it's bone. What am I doing here? That's supposed to be in. I don't know what that is. That is absolutely horrendous. We're going to get rid of that whole thing. Start again. And come from bone marrow. Marrow. I put a W in there for some reason. Specialized cells. Specialized cells. And I am not going to write down what their jobs are, but yesterday we put in our note, there was about 5 or 6. What are the different types of specialized cells in our bodies? What are types? Give me one. Muscle cells. Muscle cells. Other specialized cells. Yep. You're being even more specific than I want you to be. You're right, white blood cells, but let's just call them blood cells. All right, there's red and there's white. We'll talk more about that when we talk about the circulatory system. But I'm going to just put blood cells. But you're right. White blood cells would be a subset of that. Other ones. What other types of specialized cells do we have in our bodies? Yep. Fat cells. Store energy in the form of fat. Other cells. Yep. Skin cells. Skin cells cover our body so so all that water doesn't evaporate away and we dry out like a dried out something. Makes a difference. Other cells, am I missing any? What am I missing? What do we got? Yep. Nerve cells. Nerve cells transmit electrical signals. What have I got? 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5. Which one are we missing? Anybody see which one we're missing? Bone cells. Bone cells. All right. Oh, we also talked about the ear. The ear and the way it works, and see the note, right? They've got those little hairs inside the cochlea, and when the right frequency of sound enters and causes those bones to wiggle, the certain hairs vibrate and we hear, all right? And those little hairs called cilia vibrate in response to that. They will knock them down, yep. And actually, repetitive sounds. So let's say that someone worked in a factory for 35 years, all their career, and maybe 20 years ago they weren't very strict on hearing protection. And let's say it was like a press or something that maybe used air and it went all day long. What can happen is those same hairs vibrate all day long, and after a while they lose their ability to vibrate. And that means those people lose their ability to hear those particular sounds. Now, noises like and hitting a hammer, clang, 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 those are those really high frequencies. They're way up there. So older people tend to lose the ability to hear those really high frequencies. Now, it's interesting. The high frequencies, when we speak and say words, tend to come at the beginnings and the ends of words. So even if I use the term words, at the end, that little S sound, right, is a high frequency. So someone who's older or maybe worked at a plant like that has lost the ability to hear the end of that word, all right? And so they hear things that are muffled like this. They hear muffled sounds, and they can't hear those high frequencies. And so they say something, like you say something to your grandpa, and he says, "What? What'd you say? Why do you have to mumble? Why do you have to talk with marbles in your mouth?" Right? And so then they'll look at you, and then you'll say it again, and they do a little cheat move. They've slowly learned how to read lips, and then in the end, they can hear what you're saying, right? But they lose those ability to hear those high frequencies. Gunshots would do the same thing. So be safe. Don't have old man hearing before your time, Logan. All right. Okay, I am done there. I'm going to just stop my recording and I'm going to direct you to the textbook questions.